Hello and welcome to today's webinar hosted by KAPA Biosystems. My name is Andrea Carbone and I'm the Marketing Manager and Moderator for this event. Before we begin, I would like to briefly go over some GoToWebinar features to help ensure everyone enjoys our presentation. All attendees have been muted for the call to provide the best sound quality. However, we will be taking questions, so please feel free to type them in the questions panel throughout the presentation and we'll attempt to answer all requests at the end of the webinar and within our 1 hour allotted time frame. Today's presentation will be recorded and hosted on KAPA's website later this week, and an email notice will be sent to all attendees. So today, Dr. Mareka Appel will share insights and improvements on FFPE library construction for Illumina sequencing. In Part 1, she'll discuss quality control of FFPE samples and quality score ratios as they correlate to sequencing results using the KAPA Human genomic DNA Quantification and QC Kit. In the second part, the focus will turn to key library construction metrics using the KAPA Library Quantification Kit for libraries constructed from FFPE and how conversion rates of adapter-ligated libraries prove to be the major bottleneck in FFPE library construction. Dr. Appel will share how the new KAPA Hyper Prep kit is helping to address these issues. So Dr. Appel received her PhD in biochemistry from the University of Stellenbosch in South Africa in 2001. Where the focus of her postgraduate research was molecular plant pathogen interactions. Prior to becoming KAPA's first employee in 2006, Dr. Appel worked for a small biotech developing novel targeted cancer therapeutics. At KAPA Biosystems, she initially formed part of the research and development team, participated in establishing KAPA's technology platform, and was responsible for developing several PCR product lines. Since 2008, Dr. Appel has played a leading role in technical support and operations. She currently leads the technical team in efforts to overcome the challenges of NGS library preparation. So thank you all for attending, and I'd like to welcome Dr. Mareka Appel to begin her presentation.
Thank you very much, Andrea, for the introduction, and thank you everyone for taking the time to attend our webinar today. So as you know, KAPA Biosystems has been involving enzymes and developing kits, especially for sample preparation for Illumina sequencing for a number of years. And we've been very focused on FFPE as it's becoming a more and more important sample type in both research and clinical applications. And so hopefully in today's seminar we can provide some insights on library construction from FFPE DNA and also talk about the improvements that we've made to some of our kits for these applications. So in my seminar today I'll start with just a quick overview of the nature of FFPE DNA and the impact that the molecular damages has on library construction. Then we'll talk a little bit about FFPE DNA quality control. I'll then introduce you to two qPCR-based assays that KAPA has developed that's become very useful in our investigations of FFPE library construction. The first is the Human Genomic DNA Quantification Kit, which is used for FFPE sample QC, and then the KAPA Library Quantification Kit that's used to quantify adapter-ligated libraries. Then in the main part of the seminar, We'll look at two things. The first is the correlation between Q ratios generated with the KAPA Human Genomic Quantification and QC Kit, and how those can be predictive of the outcome of library construction and sequencing. And then the second part of the seminar, we'll look at library construction from FFPE in more detail, really identify the bottlenecks and talk about new developments to improve the quality of FFPE libraries and also sequence quality. So as all of you know, FFPE DNA is fraught with molecular damage. So we have single-stranded nicks in DNA, double-stranded breaks. So FFPE DNA just is fragmented, contains a lot of single-stranded DNA, and then inside the DNA molecules we have a whole lot of different molecular damages that includes deaminated and oxidized bases, methylene bridges, abasic sites. And these damages are a combination of the initial treatment or embedding of the tissue and then also the DNA extraction. And these molecular damages potentially affect our ability to fragment FFPE DNA consistently and with predictable success. It potentially affects the efficiency of end repair, A-tailing, and ligation during library construction, and then definitely affects our ability to amplify libraries constructed from FFPE DNA. The other consequence of these damages is that it lowers the usable amount of input DNA. So when we put 100 nanograms of FFPE DNA into a library construction pipeline, the amount of DNA that's actually available for library construction could be far less than that because of the molecular damage. And the main challenge here is to be able to process FFPE samples of variable quality in standard sample preparation pipelines with predictable success. And so the slide here summarizes pretty much, what I think a lot of people are seeing with FFPE DNA. So this is data that we've generated from, this is average sequencing metrics for libraries generated from either 9 samples. This is DNA isolated from cell line DNA, so regarded as good quality samples, versus 8 FFPE samples. And this is all libraries prepared for targeted sequencing using a custom numbleGen panel. And I think the sort of things that we see in this data is very very common to FFPE library construction in general. So firstly, the average insert size is smaller for FFPE DNA. So if you share your DNA to around 200 base pairs, for high quality DNA, we would expect the library to have an average size distribution around 320 base pairs after we've added the Illumina adapters. But in reality, FFPE libraries always end up a little smaller, and it's that sort of shrinking effect, and that makes it difficult to get certain types of data out of FFPE libraries. So in this case, on average, the libraries made from the FFPE samples were about 18% smaller in terms of average fragment size. Yields of amplified library is less. I mean, that's something that we all battle with. And so in this specific experiment, we did 8 cycles of pre-capture amplification, 12 cycles of post-capture amplification, but in the end ended up, on average with about 4.5 times less DNA from the FFPE samples from the same input, which was 250 nanograms. Library diversity is definitely affected as well. So in this case, 3.5 times less unique molecules in FFPE libraries than the libraries prepared from the higher quality DNA. And then something that's associated with this is duplication rate. So I think this is the nightmare with FFPE library construction, high duplication rate. So in this specific experiment, almost 3 times higher than for cell line DNA. So the question really is there something that we can do? Can we apply upfront QC to better predict whether FFPE samples will pass library construction and ultimately produce usable sequence data? And is there something that we can do to alleviate these effects on library quality produced from FFPE DNA? And so the question on whether or not to QC FFPE samples prior to library construction has many answers, and it very much depends on who you're talking to. So in general, I think the feedback that we're getting from labs that are process research samples is that they're going to process a sample anyway. The idea is that samples are precious and limiting, and we don't want to waste some of that sample on upfront QC, particularly if that upfront QC doesn't inform the library construction process. So if it's not helpful in saying, hey, I know the sample, has a certain quality. Because of that, I need to increase the input or use so much DNA or use so many cycles of amplification to make sure that the sample will pass. So generally, I think the viewers will process the sample anyway. In some cases, some data from a sample is better than no data at all. And then, as we discussed, if QC does contain precious sample material, it's expensive and time-consuming, and it really, especially in the research setting, is only worth it if it provides some actionable data or somehow informs the sample preparation process. On the other range of the spectrum, or the CAP-CLIA labs, or the clinical sequencing labs, in this case, samples are processed, somebody ends up paying for it, and in general, there's a much higher level of caution, and so typically samples won't be processed if they're likely to fail, and so these labs are more more amenable to doing upfront QC. And some of the feedback there is it's great to have tools to allow us to process poorer quality FFPE or FFPE from lower input samples. So typically, clinical labs will only process a sample if they know it's if they know it will succeed. So at this point, the cutoff, the quality cutoff is much higher than for research samples, and the input cutoff is also much higher. And so where in a research setting, people will still try to make a library from 50 nanograms of FFPE DNA or FFPE with a really poor quality. In clinical labs, that's not the case. And the challenge there is how can we sort of increase that spectrum of samples that can be processed successfully, and how can we predict that we can process these successfully. So most labs at least do a little bit of QC to look at concentration and size distribution of FFPE DNA. Concentration obviously is important to do some sort of a concentration determination, to decide the input or see whether the sample meets the minimum input requirements for the library construction pipeline. But generally, spectrophotometric or fluorometric concentration assays aren't really good predictors of library or sequence quality. So what we've done here is just plotted concentration. This is determined by a qubit high sensitivity double-stranded DNA binding assay of control samples. So this is DNA isolated either from fresh frozen tissue, cultured cells, or blood, so high-quality DNA samples versus FFPE DNA samples. And we plotted that against two key sequencing metrics, either mean target coverage or duplication rate. And this was again in a targeted pipeline Numbleton capture with a custom panel. And so as we can see, there's really not a good correlation for either good quality DNA or FFPE DNA between the concentration of the input sample and the outcome of sequencing. And so there's pretty much a good divider here, in terms of the sample type. So FFPE DNA on average has lower target coverage and high duplication rates. But if we look at concentration, low concentration samples sometimes have high duplication rates, but so does high concentration samples, and vice versa. Similarly, electrophoretic assays aren't good predictors of sequence quality in the end. So these are, this is just, we've given here the percentage of targets covered at 20x for a few FFPE samples processed in a whole exome capture pipeline. And so this is what we typically see for FFPE samples. Sometimes we see kind of, the DNA doesn't look fragmented at all. The electrophoretic profile looks very much like that of a good quality sample. And in many cases, we see an appreciable amount of fragmented DNA in that initial sample. But as we can see from these profiles, so here we have one where the sample looked fairly fragmented, and we got a very low percentage of targets covered at 20x. But this is a very similar looking sample, and the target coverage was actually much better and quite acceptable. So again, electrophoretic assessment upfront isn't really a good predictor of the quality of the library that one can generate from that sample and the ultimate sequence quality. So I'd like to introduce you to two KAPA qPCR based assays. Many of you are probably familiar with these, but we found these assays very, very helpful for sample QC, and then also to define key library construction metrics and help us better understand library construction from FFPE samples. So the first assay is the KAPA Library Quantification Kit, which is initially designed for the quantification of libraries either prior to pooling for capture or sequencing, or for accurately quantifying the concentration of a pooled sample prior to sequencing. But we use that kit now not only to sort of look at the final library concentration for pooling or not, but to look at key points during the library construction pipeline so that we can better understand what's happening there. And then we have the second qPCR-based kit, which is the human quantification, human genomic DNA quantification and QC kit, which is designed for assessing upfront sample quality. So the library quantification assay is designed for quantifying adapter-ligated library, whereas the human genomic quantification and QC kit is designed for quantifying input DNA. And so just briefly, the principles behind these assays, so both of these are assays for absolute quantification. They each include a set of standards, of which the absolute concentrations are known. KAPA Cyberfast qPCR master mix, so this master mix contains our engineered KAPA Cyberfast enzyme. That was an enzyme that we developed through directed evolution to be more robust, more tolerant to cybergreen. But like all our enzymes, end up being able to amplify DNA with different characteristics with similar efficiency, and that really makes it a good enzyme for quantifying libraries which are complex populations of DNA rather than single amplicons. So in the KAPA library quantification kit, and this is available for Illumina, Ion Torrent, essentially all the major sequencing platforms, we supply a primer premix, the qPCR master mix, and a set of pre-diluted standards. And essentially you amplify the standards with the appropriate primer as well as your samples. This allows you to do produce a standard curve from which the concentration of a sample is derived. And this kit can be used to quantify any Illumina library. So I'm just going to speak about Illumina today. Any Illumina library that already contains the P5 and P7 sequences. So after ligation with full-length adapters or after library amplification during which indexes are added. There's also possibilities if you're working with customized adapters or truncated adapters. There are possibilities to adapt this assay to allow you to assess the yields of adapter-ligated library before you've added the flow cell primers. And then the human genomic DNA quantification and QC kit, the principle is exactly the same. So again, there's a set of pre-diluted standards. In this case, the standard is based on a single copy human locus, or fragment of a single copy human locus. And unlike the KAPA library quantification kit, quantification is done with more than one primer set. So we have a primer set that defines a very short fragment of the single copy human locus, and then 2 additional primer sets that defines longer and longer fragments. And the principle is if you're quantifying high-quality DNA, then the copy number for these 3 primer sets is essentially the same. So if the DNA is not damaged, when you do absolute quantification with the primer set defining the 305 base pair amplicon, the concentration will be very similar to what it is when you use the shorter amplicon. And when you divide these 2 concentrations by one another, you get a ratio of close to 1, and we call this a quality score or a Q ratio. On the other hand, when you're working with damaged DNA, so any kind of damage that renders the DNA unamplifiable, so whether it's just because it's fragmented or nicked, or whether it's deaminated, oxidized, or there's other sort of molecular damages prevents amplification. What happens is as the degree of damage increases, the effective copy number for a primer set that targets a longer and longer amplicon becomes lower. And so when we determine the concentration with a 129 base pair amplicon of this standard, it will be lower than when we use the 41 base pair. And for the 305 base pair primer, it's even lower. And so when we divide the concentration obtained with this primer by the one obtained with the primer set defining the shortest amplicon, this Q ratio quality score drops below 1, and it, for very badly damaged DNA, it basically doesn't even support amplification with the 305 base pair primer set in most cases, and this ratio approaches 0. And so in the next part of this presentation, I'm going to show a little bit of data trying to use the quality scores determined with this assay to predict the outcome of library construction and inform the process. So in the first part of the study, we made libraries from 6 DNA samples that were regarded as higher quality. So they this DNA isolated either from cultured cells, from fresh frozen samples, or from blood, and then 14 FFPE samples of variable quality. And what we have here is the quality score generated with the 129 base pair primer, which we find most informative for FFPE samples. And we've ordered the samples on the x-axis in order of increasing quality, so based on this quality score. And then we've tried to correlate this with 2 major key sequencing metrics. Again, this was for targeted capture using a custom Nimblegen panel. And first we look at duplication rates. We know that that's kind of an important metric, especially for FFPE DNA. And we found actually a fairly good correlation. So, with the R-squared value over 0.8 between duplication rate and Q ratio. And this was an interesting sample. This was supposed to be a high-quality sample, but actually ended up being damaged somehow during the extraction process and behaved more, it still behaved like a high-quality sample, even though the Q score was actually a little lower than expected. The second part, we sort of looked at mean target coverage, so the inverse effect. So as sample quality increases, we get lower duplication rates, but increased mean target coverage, and still a fairly good correlation. So then we looked at another data set. This is part of a much larger data set. These are libraries prepared, from a range of DNA inputs, from 50 to nanograms to a microgram, either in whole exome sequencing workflow or custom panels or Nimblegen capture. So what this institution normally does is they use an Illumina OmniExpress SNP genotyping assay to try and predict the outcome of library construction, decide which samples should be processed for sequencing. And the cutoff is basically 72% of SNPs detected at a 0.7 call rate. And this is represented, so for this limited set of samples by the blue triangles. Green is sort of the key sequencing metric that's looked at. And typically, at least 80% of bases needs to be covered at 20x. Otherwise, a sample needs to be reprocessed and resequenced. And so we can see that in most cases, there's a sort of most of the sample is passed. In some cases, the SNP call rate was actually low, but we still obtained quite acceptable sequence data. So then we superimposed the Q ratio obtained with the 129 base pair primer set from the human quantification and QC assay, the KAPA assay, on this sample set. And again, unlike the previous sample set where there's sort of a linear correlation, we didn't really see a correlation because all samples basically passed. When we inspected the larger data set more closely, essentially discovered that most of the samples that are processed, that were processed, had a Q ratio in excess of 0.4%. And so when we went back to the other two data sets and kind of just looked at that as a sort of a cutoff point rather than a gradual correlation, it pretty much held for this data set as well. So samples with a Q129-41 ratio around about 0.4 sort of grouped together in terms of having lower duplication rates as a group and much higher target coverage. And so what we concluded from these two data sets is that FFPE samples with a Q ratio determined with the 129 and 41 base pair primer sets with Q ratio of 0.4 or higher yields libraries that has a good chance of success when processed in a standard pipeline together with high quality samples. Please remember that this applies to target capture workflows, and so the cutoff we expect for whole genome sequencing, for instance, would be a little higher and wasn't addressed in the study. So now that we've learned a little bit about predicting the outcome of library construction using our QC assay, we also wanted to dig and better understand what goes wrong during library construction from FFPE DNA. Where really are the bottlenecks, and what are the areas that we should be focusing on improving? And so just coming back to one of my initial slides again, we've discussed that molecular damage potentially affects fragmentation, our ability to repair ATAD and ligate adapters, but definitely also library amplification. And we always assume this to be one of the primary bottlenecks because we know that FFPE DNA contains a fairly large percentage of deaminated and oxidized bases, and those templates that contain these deoxidized and deaminated bases are not utilizable by high fidelity enzymes that are used for library amplification. So we know, for instance, our KAPA Hi-Fi enzyme, which has become the gold standard for library amplification, cannot amplify DNA that has a large degree of deaminated and oxidized bases. And so again, with collaborators, we set out to look at a bunch of data sets, and we extensively used our library quantification assay to define two key metrics. So normally, in sequencing workflows, people basically measure, again, the concentration of the final library. The idea of quantifying the library is just to make sure that you do equimolar pooling for either, going into capture or finally going into sequencing. But we decided that we wanted to, interrogate this process much earlier on. And so we've defined two key library construction metrics, the one we call percentage conversion or conversion rate, and that's the amount of input DNA that gets converted to adapter-ligated library. And so we typically, at the end of ligation, so after the post-ligation cleanup, we'll measure the amount of adapter-ligated library. We also measure the amount of input DNA, so that's the DNA preferably that actually goes into library construction and not into fragmentation. We know that there's a sort of an undefined percentage loss during fragmentation. So we like to just use qubit or something like that to quantify exactly how much DNA goes into library construction and then how much adapter-ligated library we get, and we express that as a percentage of input, and we call this conversion rate. And this is a really important metric because at that point you're still working with unique molecules, and whatever unique molecules you've lost at the end of adapter ligation basically is lost for the rest of the process. We can do library amplification, and we do to generate enough material for the downstream process, but during amplification we're really only just duplicating molecules. And so this conversion rate, that is the metric that ultimately determines the diversity of the library. And then, of course, amplification efficiency is important because if there's inefficiencies in that process, we may be doing too many cycles of amplification, and that affects duplication rates. And so what I'm going to present you is three sort of data sets in which we processed control DNA. So these are either commercially available human genomic DNA or DNA isolated from something like blood or cultured cells or something like that. So good quality samples and then FFPE samples in the same library construction pipeline. So this was all done with KAPA's library high-throughput or low-throughput library preparation kit, our so-called with bead protocol. That's actually very well sort of optimized, and we get pretty good results with FFPE DNA comparative to kits from other vendors. And so again, what we've tried to do is arrange the FFPE samples in order of increasing quality. And so for the different data sets, we had different types of upfront QC data available. In this specific experiment, these samples had been previously sequenced, and so we just sort of arranged them in order of increasing quality based on the outcome of previous sequencing. We had a total of 4 samples here, and in this case we processed different inputs, so 25 or 50 nanograms, and if there was enough sample, 100 and 200 nanograms. So essentially these are sort of multiple libraries prepared from the same sample. And then we measured the amount of adapter-ligated library after adapter ligation with the KAPA library quantification kit, and then also we measured the amount of amplified library. And then, we can use it. So if the nice thing about measuring adapter-ligated library is that you also know exactly what goes into the PCR reaction. And so we can use just a standard exponential algorithm to say if we start with 100 nanograms of DNA, all that DNA is amplifiable, and we have 80 or 90% amplification efficiency theoretically. We know how much DNA we should be getting at the end of library amplification. So using that number and those numbers and algorithms, we can retrospectively work out what the actual amplification efficiency was. And what was interesting in this study is that we saw conversion rates. We know with our library preparation kit is pretty well defined, and we expect something in the range of sort of 15 to 40% on average, about 20% for high quality DNA. That's sort of conversion rates that we very typically see with our kit. And then for FFPE DNA, we saw that these conversion rates were actually lower, and as the quality of the FFPE DNA decreases, the conversion rates actually decrease as well. What was very interesting is that we saw a decrease of amplification efficiency with increase of the amount of template into the reaction, and I'll come back to that in a second. So here's the second data set. Here we had 3 FFPE samples. Libraries were constructed from as close as 100 nanograms as we could, compared to, again, library construction from 100 nanograms of good quality commercial DNA. And in this case, the FFPE samples are arranged based on quality scores obtained with the human quantification kit, the human genomic quantification and QC kit. And again, we observed the same thing. So conversion rate for good quality DNA in that sort of range of 15 to 40%, actually much closer to 30% in this experiment, but much lower conversion rates for FFPE DNA. In this case, amplification efficiencies were sort of the same across the samples. But seeing again that little effect sometimes of decreasing amplification efficiency with increasing input. And then third data set told exactly the same story. So again, conversion rates for good quality DNA, in this case DNA isolated from fresh frozen tissue versus FFPE DNA. These samples were arranged in order based on quality scores and also data from an endpoint PCR assay. And the same effect again, much lower conversion rates and amplification efficiencies kind of decreasing with increasing input. So if we look at this data in summary, definitely the bottleneck in library construction doesn't seem to lie with amplification per se, but with the ability to convert input DNA to adapter-ligated libraries. And across these data sets, somewhere between 60 and 85% less input DNA ends up as a adapter-ligated library from FFPE DNA. So if you started library construction with 100 nanograms, you would be getting 60 to 80% less DNA than if you started from a FFPE sample than if you started with 100 nanograms good quality sample. Amplification efficiencies were not that different. And in fact, in one case, the average amplification efficiency with control DNA, good quality DNA, was actually a lot lower. And further analysis of the data showed that in many cases, because the library, the KAPA library preparation kit is so efficient and yields actually a high percentage of adapter-ligated library, amplification efficiency is often limited by primer depletion during the assay. And this is something that's easily been fixed, but it's something that it's worthwhile assessing, because if amplification efficiency is not as efficient as it should be, you're obviously going to get lower yields, do too many cycles of amplification, and that's just something that you should be aware of and fix right up front when a library preparation workflow is being developed and optimized for the first time. So to summarize, FFPE DNA limits significantly less adapter-ligated library than high-quality samples. And as we discussed before, it's that sort of process from end repair to ligation that really ultimately determines the complexity of the library, the diversity of the library. After that point, you're just going to create duplicates. And so lower yields of adapter-ligated library means lower library diversity. It means you have to do more cycles of amplification to get to where you need to be. And typically that's sort of in the range of 500 to a microgram DNA for capture workflows. More amplification increases duplication rates. That's so notorious for FFPE samples. If amplification efficiency is not limited by primer depletion, yes, FFPE DNA does amplify a little less efficient than good quality DNA as we expected, but the overall effect is actually much lower than we thought because of exponential amplification. And so, if you take the same amount of adapter-ligated library from FFPE versus good quality DNA, the real effect lies basically in the first cycle of amplification. So for the FFPE library, there'll be a number of molecules that's oxidized and has other deaminated, has other molecular damages that prevents a library molecule that you did manage to put adapters on from being amplified. But from the second cycle of the PCR, you basically have generated, so you're amplifying good quality DNA from the second cycle. So you lose some of the diversity during the first cycle, but from that point onward, amplification efficiency actually proceeds very, very well, even for FFPE libraries. And so what this has prompted us to do is to develop a new library amplification primer premix. This is at this point available just for Illumina libraries, where we just reformulated the primer premix to make sure that the primer concentration is high, the primers are formulated optimally, that the ends are protected against exonuclease activity, and just make sure that we don't run out of primers during the amplification reaction with KAPA Hi-Fi. But we've done more than just that. So as I said, amplification efficiency is actually a fairly simple problem to solve. But what can we do about this conversion rate? How can we manage to make libraries from FFPE DNA more efficiently? The good news is that we think that we've recently made some progress in this regard. So we've just launched a few weeks ago a streamlined DNA library preparation kit that's called KAPA Hyper Prep. And the whole idea of that project was to just, we've spent a lot of time developing a very well optimized DNA library preparation kit with a with-bead protocol to really limit sample loss, get good yields from low inputs and FFPE samples. And then we set out to just streamline the protocol because, as in many pipelines there's definitely a challenge to produce libraries quicker and reduce the overall turnaround time, especially for clinical sequencing. What we found there was when we took the cleanup steps out of our well-optimized protocol, library yields dropped a lot, and so we had to reformulate the chemistry entirely in the Streamlined kit. So we now do N-repair and A-tailing in a single tube, go to ligation, and there's only two cleanups in the end versus 5 in the standard protocol. But a big consequence of the re-optimized chemistry and the redefined chemistry is that we are actually addressing this issue of conversion rates for FFPE DNA. So here's again controlled DNA, good quality DNA, 100 nanograms processed with different kits. So the gray bars here is our standard with bead protocol. This is the same kit for which we showed you all the previous data. And you can see good quality DNA conversion rates again, right up there around 30%. And then it drops significantly for FFPE samples. So these are 3 FFPE samples, again, arranged in order from left to right of increasing Q ratios. With the Streamlined kit, we get yields on good quality DNA very similar to what we get with our well-optimized with bead protocol. But the key difference is that we're getting much better conversion rates with FFPE DNA. And so we're bringing the yield of adapter-ligated, in other words, a usable amount of DNA in that initial sample. We're almost bringing that back to what you can achieve with good quality DNA. There's huge implications. So because it means that you're getting higher library yields, it means less cycles of amplification, it means lower duplication rates. And here's just data from one of our customers that was an early access test site for this kit. So these were libraries made from 2 FFPE samples. This was for whole exome capture with a Nimblegen workflow. And we're just showing you some coverage data. So this is a percentage of bases covered at 5x, 10x, 20x, 50x, and so on. Average coverage. So 95% of bases covered at 20x for these two samples on average. Average coverage about 99x. Target specificity unaffected. But the huge difference here was the duplication rate. And so This customer reported that typically, they see 50 to 90% duplication rates with samples like this, whereas with the Hyper Prep kit, it was reduced to the range of 10 to 20%, and so obviously very excited about that. So after this, we wanted sort of to return to what we've achieved in part one. So in part one of the seminar, we said, well, we have the human genomic quantification and QC kit. It's designed to get some QC information about input DNA. And what we found is, instead of there being sort of, instead of having to tailor workflows for different Q ratios, there seems to be sort of a cutoff at around about 0.4, for, at least for target capture workflows. And if a sample has a Q ratio, the 129/41 of 0.4, you have a reasonable chance of processing that in your standard pipeline. It may not give exactly the same library quality as an equivalent amount of high-quality DNA, but it will at least pass the basic sequencing requirements. So what do we do with samples that have a Q ratio less than 0.4? So typically, if you have a low-quality FFPE sample, library yields are low, you have to do more cycles of amplification. It's actually the worst thing to do because at that point, you're just going to create more duplication. More duplication gives less usable reads and coverage drops in the end. And so for samples if we want to process samples with a Q ratio less than 0.4, really the thing that we should be doing is instead of increasing amplification cycles, we should try and increase the input into library construction. I know that that's not always possible. It's probably the most difficult thing to do. But there's also other things you can do. So if it's impossible, if you do have 200 nanograms of that sample available, don't just process 100, because the more DNA you put into the system, the more adapter-ligated library you'll get. And so the less amplification you have to do, less duplication, better coverage. If that's not possible, increase the adapter insert molar ratio. That's something I didn't really have time to talk about, but we've seen that in other low input applications, that increasing the adapter insert molar ratio does increase the efficiency of conversion, so input DNA to adapter-ligated molecules. And this is one thing that's great about the Hyper Prep kit. The optimized chemistry gives us the opportunity to play with this ratio a lot more than we are with our with-bead protocol. One small thing that we always need to do, make sure that library amplification is optimized. So if you are doing 8 cycles of amplification, make sure that every single cycle of that counts and that we're not over-amplifying and running out of primers. And then if input DNA is really limited and using the Hyper Prep kit, increasing the adapter insert molar ratio, making sure that everything's optimized, if that really just doesn't get you where you need to be with very low quality samples, try to reduce the output of the pre-capture amplification specifically. So typically people aim to make a microgram of 2 microgram of DNA, and sometimes that's what you need. If you're going to do single captures, for instance, you need a microgram of each library, and there's sometimes not more you can do. But for instance, if you're doing multiplex capture and you're multiplexing 8 or 12 or 16 samples together, you end up needing very little of each library, somewhere in the range of 100 to 200 nanograms. So don't aim to make a microgram if you don't need one. Aim to make 200 or 300 nanograms, which still gives you enough for your downstream process and also for QC. And do things like, for instance, if you need to look at your library on a bioanalyzer or other electrophoretic device, use the highest sensitivity device you can so that the QC requirement does not add to the requirement of library that needs to be generated. So to summarize, KAPA offers qPCR assays that help us to better understand library construction and does provide some actionable input for library construction from FFPE samples. So Q ratios can be predictive of sequencing outcomes. And then the KAPA library quantification kit can really be used to interrogate key library construction metrics. And we don't suggest that you do qPCR and after ligation, after pre-capture amplification, and after post-capture amplification once a production workflow is optimized. But while you're busy developing and optimizing for a specific sample type, measuring at those sort of places and really understanding how far you are from being optimal is really, really valuable, because if your pipeline is very well optimized from the beginning, it's going to be robust and it's going to have a lot of tolerance for samples of different quality. If it's very close to breaking point, like right on the edge from the beginning, it's going to break really quickly. And then you just end up having to troubleshoot and come back. So when we sort of help people develop and set up pipelines, looking at those metrics up front are really, really invaluable. And then secondly, we're very excited about the Hyper Prep kit, the fact that it does seem to address the key bottleneck in library construction. And we've seen this, in many cases now that the Hyper Prep kit does give higher conversion rates, so higher yields of adapter-ligated library compared to our existing kit and compared to competitor kits. And so translating those higher pre-capture library yields successfully through the capture and post-capture amplification means, initially that you need less amplification, so lower duplication rates, better coverage, and better sequence quality overall. And it also allows us to process lower quality and lower input FFPE samples more successfully. Finally, there's still a lot to be done. I mean, we've only, I think, scratched the surface here. There's a huge impact on how tissues are fixed for pathology, how DNA is extracted. There's a lot to be done on DNA FFPE repair. These are not areas that we're actively focusing on right now, mainly because we just don't have access to as much FFPE DNA real-life samples as we'd like to, and that's why we continue collaborating and are really grateful to the input and collaborations we have. But we're also continually trying to improve this KAPA Human genomic quantification and QC assay to see how it can better sort of predict the outcome of library construction and inform sample preparation processes. And finally, in this study, so I'd like to acknowledge the Genome Institute at the University of Washington in St. Louis, the Memorial Sloan Kettering Cancer Center, so the Bean Translational Call and Department of Pathology, Case Western Reserve University, Ambridge Genetics, and then also colleagues at KAPA Biosystems who contributed a lot to just a basic understanding of library construction parameters and have developed all these great kits that we are able to offer. So thank you very much for your attention and your questions. So we've got about 15 minutes to answer questions. A lot of the data that we would discuss today are available in posters that we presented at AGPT this year. So please contact us. Our technical email address is support@biosystems.com, kapabiosystems.com. We're happy to send you copies of the posters or discuss any of the information we presented today in more detail. And then if you want to request a trial kit, so any of the kits that we discussed here today, our with-bead library preparation kit, Hyper Prep kit, or either of our qPCR kits, please feel free to visit our website or contact your local sales representative. And with this, I'm handing back over to Andrea. Thank you very much.
Excellent. Thank you so much, Marika. We hope everybody enjoyed. I'm actually just going to put us on mute for a moment and take a look at some of the questions. A few have come in. Here's an invitation again that if you do have any questions you'd like to ask, please use your question panel, and I will tabulate them momentarily. So hang on for 2 seconds and we'll be right back. Okay, we're going to start with the first question that came in. Thank you very much again. So Marika, have you done any work to compare the Hyper Prep kit with the Illumina Nano, and are there differences you can share?
Yes, so we have done some comparative analysis for libraries prepared with both good quality and FFPE DNA. In general, what we do see with the Hyper Prep kit, as well as our existing library preparation kit, is higher yields of adapter-ligated library. When we compare libraries prepared from the same input DNA, same input amounts with Illumina's Nano kit, we think that these, the way we understand these differences. With the existing KAPA library preparation kit, the with-bead protocol is designed to limit sample loss during library construction. It's also a very well-optimized protocol, whereas the Nano protocol still follows the more traditional design where DNA is eluted off the beads after every enzymatic step, and that's associated generally with sample loss. Also, we've gone to a lot of trouble to optimize enzymatic reactions, just the efficiency of each reaction. So generally, whether you're using our with-bead protocol and then specifically the Hyper Prep protocol, we're seeing higher yields of adapter-ligated library from the available input, and that typically translates to better sequence quality. And this has been verified by a couple of large genome centers and also commercial companies.
Excellent. Okay. So the second question we have. So the protocol workflow looks simple. Are the kits automated on any of the major liquid handling robots, and if so, which ones?
Okay, so as many of you know, we've spent a lot of time in the past producing validated protocols for our library preparation kit on the major liquid handling platforms. For our existing with-bead kit, we have validated protocols available for the Biomec FX dual pod system, the Cyclone from PerkinElmer, the Bravo option B from Agilent Technologies. We have a protocol for the EP Motion, and then the Hyper Prep is a very new kit, so the kit itself was only launched a couple of weeks ago, but we are looking to automate that on all the major liquid handling protocol platforms. So again, Biomec, Cyclone, Bravo B, and then in some of the smaller platforms too. So because this is a much simpler protocol, far fewer cleanup steps and movements and handling steps, we will also have the Hyper Prep protocol available on some of the smaller instruments like maybe the NGS Express, the Zephyr. We're working with a couple of partners to try and get this protocol available on as many platforms available. Most of these methods for Hyper Prep will probably be available sort of in the next 6 to 8 weeks. So we're looking at sort of June, July timeframe for the first set of platforms, and then we'll continue to develop after that.
Excellent. So are you using KAPA Hi-Fi Uracil Plus for FFPE samples?
So that's a really interesting question, and something that we've had long debates about. So as I mentioned, initially we thought that one of the key bottlenecks in FFPE library construction is the inability of the normal formulation of Hi-Fi to copy adapter-ligated DNA made from FFPE samples that contains the aminated and oxidized bases. And so, yes, we thought we'd just solve the problem by using Hi-Fi Uracil Plus, and part of the problem does get solved. So when you use U Plus instead of Hi-Fi for library amplification, permitted that your amplification conditions are well optimized, so you don't run out of primers, you do get higher yields of adapter-ligated library. But in the end, U Plus doesn't seem to be a good solution for FFPE library construction because it fixes mutations. So if you have a deaminated base, it's going to cause a SNP in the amplified DNA. And it all depends whether that ends up, in the feedback that we've gotten is that ends up being problematic with the bioinformatic analysis afterwards. And so because we know that we can get the amplification reaction very well optimized with the normal Hi-Fi, so we really don't want to amplify the DNA that's damaged and it's going to cause SNP in the final library sequence. And so what we decided to focus on rather is trying to improve the yields of adapter-ligated library so that you get high library diversity rather than just trying to solve the problem on the amplification end.
Excellent. So do others continue to use a picogreen assay or qubit if they use the human genomic DNA quant and QC kit, and how can I use both assays to get the most info?
Yes, so many labs still continue to use a qubit or a picogreen assay. I think the one doesn't reply. So the human genomic DNA quantification and QC kit can provide absolute quantification values too. So if you just quantify with the 41 base pair primer set, we've seen a pretty good correlation between the absolute concentration that comes out of that assay and the concentration that comes out of a qubit assay. The caveat is that that's for fairly good quality DNA. So as the DNA gets more and more damaged, and that's the whole idea of the assay, that absolute value becomes lower and lower. And the problem with qubit on FFPE DNA too, so qubit works fairly well on good quality DNA, FFPE DNA, but as DNA gets more fragmented, and there's now a strong sort of suspicion that FFPE samples of poor quality actually contains a lot of single-stranded DNA and long ends that are not, that are single-stranded and not actually easy to repair. And so qubit assays that are based on the binding of a double-stranded DNA binding dye also ends up under quantifying assays, FFPEs, poor quality FFPE and pretty badly. The advantage of the qPCR assay is that it's much more sensitive than qubit and spectrophotometric assays. So just to give you an idea, the copy number range on our human genomic quantification and QC kit goes from 12 copies to about 3,000 in the assay. So it allows you to at least get relative quantification data from samples that are very low concentration or are very damaged. Yes, so people use both. I mean, we would sort of recommend it, and again, there are some limitations in terms of throughput and sample handling. But we would recommend is to use the human genomic quant and QC assay for incoming QC, so to get an idea of what the DNA sample looks like when it comes in. and then you can base the input into sharing from that data as well, and then maybe use qubit later on in the process if you want to look initially at the actual input into library construction. So after you shared the DNA, qubit's a good quick check. At that point, the DNA is there still is some single-stranded DNA. It may not be it's not a perfect assessment, but it's much quicker at that point. Qubit assays can be used for library quantification, but it's very important that libraries end up needing not to be over-amplified. They have to be amplified very, very, optimally. If you're quantifying over-amplified libraries with qubits, so typically when you see your library and it has this high molecular weight sort of bump at the end of the bioanalyzer trace, that's a sign of over-amplification, so primer depletion during amplification. That's sort of what we call, what happens during primer depletion basically is, you're trying to run amplify the DNA, you run out of primers, so you denature the library, because library molecules are all unique. If you're not, if you don't have primers to extend at that point, you end up with a bunch of, denatured library molecules. They're all unique, they're not going to find their complements again. And so what they do is they start sticking to one another with their adapter sequences, which are common to all of them, and they form these big, what we call daisy chains or tangled knots, which are largely single-stranded, molecules, and that's the stuff that migrates a lot slower on electrophoretic systems, and that's that bump that we see. The problem with all of that is that assays based on double-stranded DNA also, a double-stranded DNA binding dye also really under-quantifies those libraries. So qubit quantification can be very misleading for libraries, amplified libraries going into pooling, pre-capture pooling, or even into pooling for sequencing. If the libraries are over-amplified, because you're going to be underestimating them, or amplified to different extents. So if you're quantifying libraries that are optimally amplified versus, and you're having the same pools over-amplified libraries, the evenness of pooling is definitely not going to be accurate. I hope that answers the question.
Okay, excellent. All right, so do you have any data showing the utility of the Q ratio with PCR-based enrichment rather than capture hybridization?
So we have worked with a few groups that have used the human the Q ratios basically in amplicon-based workflows as well. There's especially one group, and we've recently released a customer profile on that, Bob Daber at the University of Pennsylvania. So they've used the assay very effectively in a amplicon-based assay where they looked again at input DNA and then used the Q ratio to determine the amount of input DNA for the amplification, for the upfront, the multiplex PCR amplification reaction, and showed a very strong correlation between the sample quality, the Q ratio, and then the input into the reaction. So essentially, they would just use the human quant kit, get the Q ratio, and then based on the Q ratio, decide how much template DNA needs to go into the initial reaction.
Excellent. Another question is, does adapter quality impact percent conversion rate?
Very much so. So, all of what we talk about adapters, we're always assuming that all adapters in an adapter solution is fully double-stranded. So as you know, the Illumina Y adapter has a very short double-stranded segment, so that the sort of the base of the Y, it's only 12 base pairs compared to another 48 odd bases on each oligo that's single-stranded. That's the full through-seq adapter. It's not a very stable molecular structure. So if you have adapters that are not fully duplexed during adapter ligation, only those that have a fully duplexed Y at the time of adapter ligation will be able to participate in library construction or in that ligation reaction. So we talk about adapter to insert molar ratios. We've found with all our kits that it's necessary to use an excess of adapter to make sure that you get most molecules with adapters on both ends. If you're using a very low adapter to insert molar ratio, then you end up with molecules predominantly with a single adapter. So if you're working with an adapter stock where most of your adapter can't be ligated because it isn't duplexed at the time, then it's going to affect conversion rates significantly. And we provide some guidelines. So essentially, when you're buying oligos to make adapters, so either buy your adapters from a reputable vendor or buy oligos that are HPLC purified. That means they have high molecular purity. And then it's very important that duplexing is done in an appropriate salt solution to make sure that the adapters stay duplexed as much as possible during library construction, especially for high throughput systems where adapters often spend an hour or two on deck without being cooled, and we do that regularly. But yes, the adapter quality plays a huge role in conversion rates.
Okay, we have time for maybe one or two more questions. How exactly does the Hyper Prep cause the higher conversion rate?
I would say that's the million-dollar question. I don't think whether we understand all of it. We know that it has something to do with the modified chemistry, but we don't know the exact mechanism. So this is, the higher conversion rates are things that we've observed very, very consistently, but we don't know the exact molecular mechanism. So we think it's a combination of just the way we've formulated the chemistry, the fact that the protocol has a higher temperature incubation step, that there's a theory that may contribute to the higher conversion rates, and then just generally less handling and less cleanup steps may help with that. So just losing less DNA along the way, and then typically with the Hyper Prep kit, because of the modified chemistry, we use higher adapter insert molar ratios, and so we can go a lot higher with the Hyper Prep kit versus our three-step kit before adapter dimers become so predominant that we can't clean them up anymore. So just for example, with our with-bead protocol, we recommend a 10-to-1 adapter insert molar ratio for good quality DNA. For FFPE quality, DNA, we say double that at least, so work at 20-to-1, maybe up to 40 or 80-to-1. But at that point, we start just making so much adapter dimer with the three-step chemistry that we can't clean it out properly. With the Hyper Prep kit, people have gone up to, adapter insert molar ratios in the hundreds and even up to 1,000-to-1 without getting, an issue of having so much adapter dimer. So the chemistry basically inhibits the adapter dimer formation as well. So it's a combination of reformulated chemistry, a different protocol, the fact that we can use higher adapter insert molar ratios, and then less handling and less cleanup steps. That's pretty much what we think.
Okay, one last question, which is, will Hyper Prep kit work with Agilent Haloplex exome capture kit?
We've not looked at Hyper Prep and Haloplex at all. I need to think a little bit about the mechanism, and please send us your email address so that we can follow up on that question. We do know that it will be compatible with the SureSelect kits, but Haloplex, I have to think about a little bit, and we'll get back to you with a more detailed answer on that question.
Excellent. Well, thank you everybody so much. We will have, I saw a couple questions that came through. This webinar has been recorded, and then I did notice a couple comments that said that the sound was going out every now and then. So we'll listen to the webinar and see if everything is answered. There are quite a few questions that came through that Marika did not have the chance to answer, so we will pull those together as well, and within the next few days try to get a response to all of them in our follow-up email. Again, thank you everyone for joining us. Please visit kapabiosystems.com if you'd like to trial any of the kits that we've discussed today, and we look forward to our future webinar next quarter. Thank you very much.